Hello students, welcome to EPG Pathshala. I am Dr. Zinat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy in Jamia Hamdard. Today we are to discuss the module Nasal Drug Delivery Systems, Conventional Devices and Novel Drug Delivery Strategies for the same. This is from the paper NDTS 1. So students, let us try to find out what we are going to learn in this module. Beginning with the introduction, we will move on to understanding the various conventional nasal drug delivery devices. The commonest of the two, we will try to understand in detail the commonly available nasal sprays and the nasal drops. And the major portion of the module would be devoted to understanding the various novel strategies which have been experimentally used and which have been successfully used for the nasal drug delivery approaches. If we just try to understand, very commonly it is being noted that the nasal drug delivery is most commonly used for the local delivery of the drugs. The category of the drugs which are most suited for such delivery include the commonly used decongestants, antibiotics, mucolytics, and most of the drugs are probably there only for the loco-regional effect. So what we can say is that the drug devices like sprays and your drops are focusing on the loco-regional delivery of the drug through the nasal cavity. But it can also be very well exploited for the systemic delivery of drug, but limiting itself to the drugs which has got low molecular weight, much less than 500 Daltons, and which also include the therapeutic peptides. We will be now discussing about one of the commonest devices which is used for the nasal drug delivery, and these are referred to as your nasal sprays. Nasal sprays contain therapeutically active drugs which are dissolved or suspended in solutions and contains a mixture of excipients including preservatives, viscosity modifiers, emulsifiers, buffering agents, and contained in non-pressurized dispensers which has the capability to deliver a spray containing a metered dose of the active ingredient. The dose can be adjusted by the spray pump or by a pre-metered pump during manufacturing procedure. These can be designed for unit dosing, which we refer to as the single-used nasal spray devices, or can discharge up to several metered sprays of formulation containing the drug substance. Nasal sprays are applied to the nasal cavity for local or systemic effect. Commercially, nasal sprays are available in the form of squeeze bottles, which usually do not deliver the required doses. These are also available as metered dose devices, which deliver the required and fixed volume for the medication of the patient. The formulations are aqueous-based and do not contain any kind of propellant. These are necessarily sterile, which are intended for delivery to the lungs by oral inhalation for local or systemic effects. Nasal sprays may be unique in many aspects, which may include container closure system, manufacturing processes, stability of the active ingredient, controls of critical steps, intermediates, and drug product itself. These aspects should be critically considered during the developmental phase, as minute changes can affect the ability of the product to deliver reproducible doses of the incorporated medicaments to patients throughout the product shelf life. We will now discuss some of the various unique features which are elicited by the nasal sprays. Coming to the first aspect, the metering and the spray producing pump mechanisms usually include the jet and orifice design and the components that are used for reproducible delivery of drug formulations are constructed of many parts of different design formats which can precisely control in terms of dimensions and composition. The second important unique aspect of the nasal spray devices are that energy is required for the proper and adequate dispersion of the formulation in the form of a spray. This is primarily performed by forcing the formulation through the nasal actuator and its orifice. The formulation in the container closure system primarily includes a container, number two, a closure, pump, collectively constitutes the drug product. The design of the container closure system affects hugely the dosing performance of the enclosed drug product. The container closure system designs nasal spray drug products include both pre-metered and device-metered closures using mechanical or power assistance or energy from patient for production of the spray. Pre-metered ones contain previously measured doses or a dose fraction in some type of units which are subsequently inserted into the device during manufacture or by the patient before use. Typical device metered units have a reservoir containing the formulation sufficient for multiple doses, which are usually delivered in the form of metered sprays by the device itself when activated by the patient. My dear students, we would now go into the understanding of another important type of formulation design that is called as your nasal drops. The nasal drops requires the installation of one or more drops of the drug solution either from a dropper with a flexible rubber or directly from a squeezable plastic container directly into the nasal cavity. These formulations deposit entirely on the nasal cavity, which provides an adequate surface area for immediate absorption as compared to the drug delivered in the form of a spray. This indicates that some drug is deposited on the ciliated regions of the mucosa and is hence immediately available for clearance, while a proportion of the dose may be deposited at the nasopharynx where it may be swallowed up and therefore not available for absorption from the nasal cavity. This condition leads to varied absorption profile for the drugs with a narrow therapeutic window and appears to be inconvenient to most of the patients. When administered, approximately 40% of the administered dose is cleared rapidly within 20 minutes, followed by a second slower phase of clearance. In this second slower phase, clearance of the drops is much faster. Due to this faster clearance mechanism, nasal drops are more suitable for drug moieties which are rapidly absorbed in the nasal cavity. Students, it's an important point to understand that all the drugs which are being used for the nasal drug delivery are not of the same type. They might be different by virtue of the molecular weight, by virtue of their chemistry, and many other ways. It is a common understanding that the conventional drugs which are usually administered for systemic delivery through the nasal cavity are of relatively very low molecular weight, which is usually less than 500 daltons. But many a times, The nasal drug delivery is being employed for delivering the peptides and proteins which generally have a high molecular weight to the tune of 1000 Daltons and above, and therefore these are unlikely to be absorbed across the nasal mucosa in any appreciable amounts without pharmaceutical intervention. So it becomes important that in order for better drug delivery through intranasal mucosa, and particularly of high molecular weight drugs, there needs to be certain formation or devising of new technologies which can help in better drug delivery. Therefore, we have developed a large number of strategies which can be employed for the enhanced drug delivery through the nasal mucosa. Up till now, we had been talking about the various conventional systems which have been used for ages for the purpose of nasal drug delivery. There has been a continuous research ongoing, and there are a lot of studies which are being conducted in order to find the suitability of the drugs for delivery through the nasal cavity. Needless to say that there are always a large number of drugs which are present and being designed which have got very high limitations for not to be delivered through the first route of choice, that is the oral administration. The drug primarily has got hepatic first pass metabolism, it has got some stability issues, therefore we have to look for an alternative route. We have chosen an alternative route like the nasal drug delivery. The point is that the commonest means of nasal drop and nasal sprays are usually not very successful in delivering all sorts of drug. So there has been a continuous efforts on finding that what can be the various novel strategies. It is continuously being exploited, and we can in the next few minutes we are going to sort of have a capsule understanding of the various strategies in the nasal drug delivery. The first point of discussion is that we can simply go in for a strategy where we increase the permeability of the nasal epithelium. A number of molecules have been studied by various researchers which probably need to be passed through the nasal membrane, as I just probably discussed, and this does not cause noticeable harm to the nasal cavity per se. The mechanisms of absorption promotion of these molecules are many, and one of the mechanisms is that we enhance or we increase the nasal absorption process. How do we go about it? The first mechanism or the first approach is alteration of the mucus layer. There are a large number of excipients of the category like anionic surfactants and cationic surfactants, and we have got the bile salts in such categories which are known to decrease the viscoelasticity of the mucus. When the viscoelasticity of the mucus is decreased, there will be a very pronounced increase in the nasal absorption. The second important strategy probably is being exploited, and that is the alteration of the tight junctions. We have got agents like the EDTA bile salts which are able to sequester the available extracellular calcium ions which will cause the tight junctions to open. Once these tight junctions are opened, the permeability or the natural permeability of the nasal mucosa is compromised, and it will allow the drug to pass through that. As a result of this, the paracellular route becomes leakier, permitting the increased absorption of the substances that use this route. Another strategy which can be employed for the enhancement of the drug delivery through the intranasal mucosa is the reversed micelle formation technique. In case of reverse micelles, the hydrophilic surfaces of the molecules face inward while the hydrophobic surfaces of the molecule face outward from the lipidic environment. Hence, The formation of reverse micelles within the cell membranes creates an aquospore through which the drug moieties can easily pass. The unique adjuvant properties of various bile salts species effectively relate to their differing capacities to penetrate and self-associate as reverse micelles within the membrane. This uniqueness of the various bile salts make them very suitable ingredients and excipients for designing and developing of newer strategic drug delivery systems for nasal delivery. Another technique which probably has been exploited for the enhancement of the intranasal delivery is the procedure referred to as extraction by co-micellization. Solubilization of the cell membrane lipids is possible by the removal of the cholesterol by surfactants such as bile salts and polyoxyethylene ethers. This strategy can result in the formation of the dosage forms which can easily be penetrable into the mucosal membrane. There is another technique which probably is referred to as the erosion of mucosal surface, which can be very successfully employed for delivery of the drug transnasally. Erosion of the mucosal surface, loss of cilia, and the formation of a dense mucus coat as a result of injury can be caused by surfactants such as bile salts. A serious drawback associated with the use of penetration enhancers may be due to their negative effect onto the epithelial tissues, either directly by perturbing the vital cell structures or functions, or indirectly by effectively permeabilizing the epithelium and therefore creating the way for inward penetration of toxic agents as well as microorganisms. It is generally believed that when the absorbing membrane is damaged, the surface active compounds only enhance penetration. Patients have experienced stinging in the eye and excessive lacrimation when STDHF was used as a penetration enhancer at a concentration of as low as 0.8% weight by volume. This observation limits the clinical development of such compounds. It was also seen that the co-administration of cyclodextrins or phosphatidylcholine has been known to reduce the toxicity of certain kind of surfactants. In addition to exerting effects on the nasal epithelium, penetration enhancers may also promote nasal delivery via solubility effects. Cyclodextrins are used to solubilize different drugs and thus increase the concentration of the diffusion of the drug at the intended absorption site, which is supposed to be an added benefit of having the drug at a higher concentration, and this is at the same dose which can be achieved in a smaller volume of solution. For example, the addition of beta-cyclodextrin to dihydroergotamine can enable the increase of the drug concentration from 4 mg per ml to 10 mg per ml. As a result, the large volume which are usually administered to the nasal cavity can result in spillage, can be effectively reduced. Cyclodextrins are capable of dissociating the insulin hexamers into smaller form of aggregates, which may provide an additional form of mechanism for the promotion of its absorption. There exists a direct relationship between the extent of absorption enhancement by cyclodextrins and damage to the nasal membrane. Penetration enhancers may also promote the nasal delivery by increasing the stability of the drug due to the enhancer decreasing the activity of enzymes which may degrade the drug. Another technique which can be employed for the enhancement of the intranasal drug delivery is the enhanced contact time at the absorption site. The bioavailability of the drug is enhanced when the contact time between the drug and the absorption site is enhanced. As drugs are usually cleared from the nasal cavity by mechanisms such as mucociliary clearance, swallowing, or by metabolism, the inhibition of these clearance mechanisms should result in increased absorption at the intended site. There are certain strategies which can be followed to get this done. The first amongst the strategies is the modifying the actual site of deposition. An area of high mucociliary clearance in the nasal cavity is located in the highly ciliated middle and posterior region. Hence, the drug deposited in the anterior region of the nasal cavity are expected to be cleared less rapidly and hence have a tendency to be absorbed well. The contact time of the formulation with the nasal cavity by increasing the viscosity of the solutions administered to the cavity of the nose, and the agents which are required to get that enhanced viscosity are primarily polymers like methyl cellulose and hyaluronan. Higher viscosity solutions produce a more localized deposition in the anterior portion of the nose, which is the low clearance site. Localized deposition of the administered formulation may be due to viscosity related changes in the particle size of the delivered droplets. The bioavailability of the drug is also influenced by the delivered volume of the drug solution. For example, the bioavailability of desmopressin was nearly doubled when it was delivered as a 250 microliter actuations from a metered nasal spray in comparison to the delivery of 100 microliter actuation. This may be attributed to the prolonged retention of the dose at the administration site. The second strategy which is being proposed is the use of bioadhesives. Bioadhesives or mucoadhesives adhere to the biological membrane such as mucus or the tissues. Bioadhesives are known to influence the drug bioavailability by decreasing the clearance rate of the drug from the absorption site, thereby increasing the time of absorption. Secondly, by increasing the local drug concentration at the site of adhesion, protecting the administered drug from dilution and hence possible degradation by nasal secretions. Another strategy which can be very clearly employed for the enhanced drug delivery is the decrease in the rate of mucociliary clearance. The retention time of a drug is increased by reducing the mucociliary clearance rate of the drug. This effect can be achieved by including an excipient in the formulation with a reversible celiostatic effect. Such kind of agents which can be used can be from certain groups of the preservatives. Many penetration enhancers which cause irreversible celiostasis are STDHF, which is used in the concentration of as low as 0.3%. Then laureth 9.3% concentration can be used. Sodium deoxychelate again in a concentration of 0.3%, or irreversibly halt the mucociliary transport, and which is done by an increased concentration up to 1% of STDHF and 1% of laureth 9. A number of bioadhesive formulations which probably can be of help for enhanced retention are summed up as: number 1, the bioadhesive solutions or suspensions. A number of viscosity enhancers are considered to be bioadhesive and putative bioadhesive polymer gels, which include methyl cellulose, sodium carboxymethyl cellulose, chitosan, carbopol 934P, and these are able to decrease the rate of mucociliary clearance in the rat by 5 to 57%. By decreasing the ciliary motility, the rate of clearance of the drug from the nasal cavity is reduced by many folds. For example, a nasal solution formulation formulated using chitosan is shown to be less toxic to ciliated epithelia than that of the STDHF and resulted to yield in an absolute bioavailability of approximately 31% for the analgesic morphine 6-glucuronide in sheep. In addition to the observed effect, chitosan has been shown to enhance the nasal absorption of insulin, which has got a huge molecular weight of 5.8 kilodaltons in rats and sheep. Some bioadhesives, such as carbomers, have also been shown to be able to form complexes with mucus, thereby increasing the viscoelasticity of the mucus and reducing its clearance rate. Some of the polymers are termed as thermogelling polymers, and these are exemplified by ethyl hydroxyethyl cellulose and pleuronic or polaxomer 407. It is pleuronic F127. Above a certain concentration in aqueous solutions, such systems are liquid at a room temperature and below, but at physiological temperatures ranging between 32 to 37 degrees centigrades, the viscosity of the solution increases. Once administered in the nasal cavity, the viscosity of these solutions increases due to the increased temperature, and the contact time between the drug and the absorbing membrane is enhanced as compared to that of the simple solution. Such systems have also been investigated to enhance the vaginal and the ocular drug delivery. Another formulation approach based on the bioaddition is where we are able to formulate dry powder adhesives. This is a different approach to deliver the active drug in a dry powder carrier system. For example, microcrystalline cellulose, hydroxyethyl starch, cross-linked dextran, microcrystalline chitosan, carbomer, pectin or alginic acids are employed. The polymer used absorbs the water when it comes in contact with the nasal mucosa and swells to become a viscous gel, which shows bioadhesive properties. Such systems can remain in the nasal cavity for long duration of time, as long as 6 hours. For example, the bioavailability of the somatostatin analog octreotide in rats was found to be enhanced by the co-administration of alginic acid and cross-linked dextran as dry powders. Certain carriers also prolonged the time during which therapeutic plasma concentration of drug is maintained, effectively providing sustained release of the said drug. This is known to occur due to the rate and extent of water uptake being modified by the formulation as well as to the type of gel formed by the excipients. As the polymers hydrate to some extent by withdrawing water from the secretions of the nasal epithelium, localized changes and mucociliary clearance occurs due to the presence of a hydrating polymer and potentially due to induced alterations in the viscoelasticity of the mucous gel. Another strategic approach to have enhancement in the drug delivery through the nasal mucosa is the approach where the inhibition of the enzymatic degradation is done. We are well aware that another form of clearance mechanism which is involved within the nasal cavity is the enzymatic degradation of the active drug substance by the nasal secretions and mucosa. For example, cytochrome P450 dependent monooxygenase metabolizes the nasal decongestants such as nicotine, cocaine, as well as progesterone. Protease inhibitors such as B-statin, diprotonin A, and aprotonin are known to inhibit leucine aminopeptidase, dipeptidyl peptidase, and trypsin respectively. Some enzyme inhibitors are active against more than one peptidase enzyme, such as leupeptin inhibits both cathepsin and trypsin. The choice of enzyme inhibitor in the nasal cavity also depends upon the peptide. For example, the inhibitors having a trypsin inhibitory effect are also known to enhance the nasal absorption of salmon calcitonin in the rat model. The compounds having penetration enhancing ability at the absorbing membrane are also known to decrease the metabolism of certain peptides. By denaturing leucine aminopeptidase and preventing the enzyme substrate complex formation, the bile salt sodium glycocholate has been known to protect insulin from proteolysis in the rat nasal mucosa. We have just discussed the various strategies which can be very well used for the enhancement of the absorption of the drug through the nasal mucosa. There are another set of miscellaneous approaches which can be of great help in context with the enhanced absorption of drug to the nasal cavity. The first amongst them is the approach which encompasses the altering of the osmotic pressure or tonicity of the formulations. It is a well understood fact that the variation of the isotonicity increased the absorption of salmon calcitonin, which has a molecular weight 4.5 kilodaltons, by 4 to 5 fold by one group. But altering the tonicity of the formulation had no noticeable effect on the absorption of human granulocyte colony stimulating factor, which has got a very high molecular weight of 19 kilodaltons. Another group in a research study observed decrease the pH of the formulation will enhance the drug absorption. Therefore, conclusively speaking, alterations of pH and osmotic pressure beyond a certain range may result in the damage to the epithelium and hence increases the permeability of xenobiotics. We also have another approach where we can talk about the delivery of the drug in the powder form. The powder form of the drug is a method which will increase the drug absorption in the nasal cavity but without the inclusion of a bioadhesive carrier. For example, freeze-dried insulin is known to be absorbed well in the form of powder than in the solution form. but the absorption of glucagon and dihydroergotamine was found to be almost same when delivered as solution or dry powder form. My dear students, this brings us to the last section of the today's module. Let us try to summarize what we have just learned in this module. One thing which is a take-home message is that the nasal drug delivery can primarily be of two varieties. We can use it successfully for loco-regional delivery of the drug, but we can very well use it even for the systemic drug delivery. The drug delivery devices can very successfully be used for delivering drugs like decongestants, antibiotics, mucolytics on one hand, but it can also be used for complex molecules like the peptides, which are needed to be delivered systemically. The most common of the devices which are being available and marketed are the nasal sprays and the nasal drops, but of course they have got a large number of limitations, and one of these is the faster clearance. The current research in the area is focusing on overcoming these limitations of the conventional devices by using a variety of approaches to increase the bioavailability of the drug. This can be increased by an exploitation of a series of strategies. We have to increase the permeability of the nasal epithelium. We can do this by simply using certain excipients which can probably compromise the epithelial permeability and then allow the drug to go. We can also go in for the usage of a very common concept of bioadhesive or bioadhesion. Herein, what we are going to do is that we are enhancing the retention time of the dosage form on the mucosa by using the variety of bioadhesive agents, and then we can further move on to a little more sophisticated methodology where we can even try to inhibit the enzymatic degradation of the formula. And all these strategies can be used singularly, can be used in combination, but with a single focus on enhanced permeability and better bioavailability of drugs through the proposed nasal route.